So we're going to get started here. We had a couple of kind of things to finish off on topic number 9. We were talking about disinfectants, and we're going to talk about some methods for evaluating them. And you're going to see that actually there's some parallels when we talk about antibiotics, right? We can use some of the same methods as well. So I know I had kind of quickly mentioned this one here. This is the in-use test, and this is basically where you test it wherever you're trying to use it, right? And maybe you've tested the old disinfectant, and then next week you try the new one, maybe a swab later and see how effective it is. Not a very good kind of really scientific test. It's hard to do consistently in terms of what you're doing, but sometimes it sort of needs to be done to see if it's actually working in the environment for which it's designed, right? I know there's some questions at the beginning of the COVID pandemic in terms of, you know, the virus, how stable it was on clothing or surfaces and all those kind of things. So somebody had to do the test, and there's actually been a couple of studies on that kind of thing with the coronavirus, right? You know, the numbers may not be perfect, right? You know, how long is it lasting on your clothes? I think it was like a couple of days, but again, you know, at least it gives you a ballpark compared to on, let's say, a surface or something like that. And then an idea of what methods might work. I know somebody was talking about, I don't even know what it was. The beginning of the pandemic, there's somebody talking about putting these things in our offices that I don't even know what they was talking about, ionizing the air or something. He kind of lost me. I don't think he knew what he was talking about. But there's probably, I'm sure there's 1,000 products out there for these kind of things. So if you want to be a little bit more scientific, what we want to do is a dilution test. And there's a few different ways to do this, but the standard dilution test for disinfectants is using the little metal rings that you can buy. I have no idea why somebody decided on metal rings, but somebody did it a long time ago, and now it's kind of the standard. And once in a while, you might see things like a phenol coefficient, meaning it's comparing the disinfectant to phenol. Phenol is really strong, right? So it's kind of a gold standard, except for it's like it'll burn your skin and smells bad. So we want something as good as phenol, but without the bad parts, right? Is often what we're looking at. So generally what they do is, you can see it says here, these are dipped in the test bacteria. So I've never actually done this test. I don't know whether you stick it in for a minute or two, but the whole idea is you stick it in and then you pull it out and you just let them dry. And usually what you're doing is having some different standards, right? So we have a gram positive and a gram negative, right? So E. coli is gram negative, Staphylococcus is gram positive. Sometimes Salmonella, right? Salmonella tends to be a little nastier than E. coli. Both of them, in terms of their susceptibility to disinfectants, are kind of pretty similar. In some ways, they're kind of similar organisms. And then we throw a wild card in there. So Pseudomonas, I've mentioned a few times. We're going to talk about it kind of a little bit later on in the semester, but it's one of these ones that it's very naturally resistant to things, right? So it's kind of a tough bug, I guess you could call it, right? It's just got enzymes, and it has these efflux pumps that pump out chemicals it doesn't like. It's got a bunch of mechanisms that makes it a little bit harder to treat. So like I said, it's kind of the wild card. It's another gram negative, but a lot tougher than E. coli or Salmonella. So that's the first step. And then what you do is you throw the rings in the disinfectant, and you can have different concentrations, right? So pretty simple. You have concentrated, you dilute it a bit, and you can test out. And then this way you can find out, what concentration is effective, and you can compare it, right? So you can see they're using micrograms per mil. So maybe one disinfectant is effective at 8 micrograms per mil. This one is, that's how you read it, where it's all clear, means you're killing all the bacteria. Not effective at 4 micrograms per mil. But maybe disinfectant B is really good at like 2 micrograms per mil. So you know that this one is really potent. And you can compare these numbers. And like I said, it's a very quantitative way to looking at things. You can compare them all for gram negatives. You compare them all for gram positives. Kind of depends on what you're trying to do. If you're looking at, treating staph infections in rooms, then you're more concerned about the Staphylococcus numbers and so on. Sometimes this is called an MIC, so minimal inhibitory concentration. That term is used a bit more for antibiotics than it is for disinfectants, but it's the same test, right? You do a dilution series and does it kill the bacteria, yes or no, right? So like I said, you get a very quantitative number, and then you can, for comparison. So that's a little bit more laborious of a test. This is not working for me here. Let me try this. There we go. There's another way to do these kind of tests that's a little quicker and dirtier. And if you ever been to a science fair, there seems to always be one kid doing this kind of test, right? And so you can test disinfectants. So maybe the kid is comparing mouthwashes, or sometimes, you know, garlic versus something else. But you can test all sorts of things this way. What you do is you get a little filter disc, you stick it in the disinfectant, and then you place it on a plate where you've put a lawn of bacteria, right? So this is called a disc diffusion test, or Kirby-Bauer test, because I guess Kirby and Bauer are two people who pioneered it. And it goes on the plate, and so you can look at the inhibition zone around the disc. So this is a little quicker and easier, and you're still getting, in some ways, quantitative results. If you have a big zone of inhibition, it means the disinfectant tends to be better. If you have no zone of inhibition, you can see Pseudomonas here. I mean, that didn't work, that didn't work, that didn't work, the chlorine did, right? So I was telling you, this one's a little tougher than your typical microorganisms. You can see E. coli is tougher than staph, and that tends to be the trend. So remember, gram negatives, they have that special cell wall with an extra membrane. That's kind of like a little extra layer of protection. They tend to be a little bit more resistant to these things. Whereas gram positives, not quite as much so, right? So like I said, another way to kind of do some quantitative measurements, and a little less laborious than, you know, making a whole bunch of test tubes and those kind of things. I can test, you know, like 4 on a plate, 4 different disinfectants, right? Or maybe 4 different concentrations, or whatever I'm trying to look at. But yeah, if you ever go to science fairs, just take a look. You'll see there's usually at least one kid doing this kind of thing, and they're comparing mouthwashes or something like that. Very easy to do. Okay, so the midterm. Let's talk about the midterm for a minute. We're, I think, a week and a half away, so November 2nd or something like that. I can't remember the exact date. Does that make sense? November 2nd? I think it's a Wednesday. I'm a little further ahead than I thought we were going to be, because I wasn't sure whether we'd have classes on the day of reconciliation or not. So I put the midterm a little later, but I'm sure everyone's fine with that. I'm not going to move it. Okay, it means you have a little extra time, but I'm going to talk about the midterm. I'm not sure if we'll get to it on Friday. Definitely next Wednesday if I don't get to it on Friday. Okay, and it's going to cover after topic 6. So topic 6 was on viruses. So topic 7, 8, 9, and 10. So 4 topics. So we haven't talked about topic 10 yet, but we're going to get there in a minute. On the midterm, there will be a question, something like this. I'm going to give you a scenario, right? A scenario where you have to disinfect or sterilize something, right? And so all of those methods we're talking about, right? Autoclaves, disinfectant chemicals, UV, and so you're going to answer the question, what you think is the best way. And then this one here, you can see there's an example. This is actually asking for 3 methods, right? And, you know, there's not necessarily one right answer. For this one here, we've got lots of disinfectants to choose from. The right answer is probably not autoclave the waiting room. You might kill the people in there, right? But autoclave is often the gold standard, right? And make sure you take a look at the wording, right? Sterilization versus disinfection, right? Those are 2 different things. Disinfection usually means you're applying a chemical. You may not kill everything. Your technique may be better than someone else's, and so on, right? So like I said, it'll be some sort of scenario relevant to a hospital, and you'll have to explain yourself, right? Okay. Can't remember what I had last year. I think last year it was some surgical equipment. So the right answer there was autoclave, right? But then that's one answer. I'm asking for like 3. So maybe number 2 would be UV. Number 3, you could maybe, you know, if you're desperate, boil it. That may not kill all the endospores. Probably a better method would be soaking it in ethanol. Ethanol is a very good disinfectant, and it evaporates afterwards, right? So there's a few ideas for you anyway. Any questions about that? Okay, so be warned, that's coming up. Okay, so that's it for topic 9. Topic 10 is going to be a couple of lectures. Let me see here. I just got to load it up. think that's sharing. Okay. So topic 10 is on antimicrobials. So tons I can say about this one. I've done, this is an area I've done a lot of research in, and there's lots that could be said, particularly about penicillin. We're going to definitely talk a lot about penicillin. And unfortunately, lots of new names. Okay. I guarantee you this will not be half as bad as your pharmacology course. One thing with drugs is that the names, you know, sometimes they're kind of make sense. Other times they're just random letters mashed together. And that's exactly what the pharmaceutical companies do, right? They can't use an existing word, right? You know, for Viagra, they can't say erection pill or something like that, right? So sometimes they're creative. Viagra, for example, they're using, you know, it sort of sounds vital. And so, but they're using a non-pre-existing word. They have to take a look and make sure it doesn't exist in any languages. And so that one, there's a suggestion that you might have more vitality or something, right? Sometimes they're just mashing letters together. Acetaminophen, yeah, just rolls off the tongue, right? But some of them don't roll off the tongue. So unfortunately, lots of words to learn. I'll try to focus on the examples that I think are most important. So first, I want to give you this one. This one's been floating around the internet for a while. So it's kind of a cute little history. You'll see it's kind of a joke too, right? So there we go. 2000 BC, here, eat this root. 1000 AD, that root is heathen. Here, say a prayer. Okay. 1850 AD, the prayer of superstition. Here, drink this potion. 1920, that's snake oil. Swallow this pill. 1945, that pill is ineffective. Let's try penicillin. Okay, now we're getting somewhere, right? 1955, oh no, they've mutated. Try tetracycline. Okay. 1960 to 1999, we've got oops. We got lots of resistance. And then that brings us to 2000 AD. Here, eat this root. So anyway, we're going to talk about resistance. I don't know if we'll quite get there today, but this is a huge topic, right? And kind of terrifying in some ways. So there's a lot to say about it, of course. So before we get too far, let's look at some definitions, right? Pharmacology is just, you know, the drug action in the body includes many things, right? It includes, you know, how does the drug do what it does? It includes side effects. It includes what tissues that drug might permeate to. It includes how long it lasts in the blood or other tissues. You know, there's a whole bunch of things that could be said about pharmacology. Sometimes we use the word chemotherapeutic agent, and that doesn't necessarily mean cancer, right? It could mean a drug treating a disease. So insulin, right? Used to treat type 1 diabetes. That's technically a chemotherapeutic agent. Antibiotic. This is the term that generically, when people say it, they usually mean a drug that will kill bacteria. It doesn't have to be, though. That's kind of the general usage of it, right? So if you have a viral infection, the right answer is you don't need an antibiotic, right? In most general terms. But technically, it could mean killing any pathogen. But most people, like 98% of the time, when they use the term, they mean an antibacterial drug. So for clarity, particularly in this unit, when I use these words, I'm going to try to be more specific. I'm going to say an antiviral drug or an antiviral, right? Antibacterial drug, because I don't want to mix this up. But if I do slip and say it once, I do mean an antibacterial drug. Okay, so some definitions for you, right? So let's go way back, right? You know, what were we doing 120 years ago, or well, 100 years ago, right? For that matter. I found this stat. I found it in a couple of places. So people were getting sick and they were dying. What do you do? Well, there wasn't much to do in many cases other than hope the immune system will deal with it. Maybe you treat the symptoms. You know, if they have a fever, you cool them down. But if you have a raging infection, in many cases, it is a death sentence, right? And so here's the stat, right? 1/3 of children dying before age 5, right? Children in particular are very susceptible. They're young, vulnerable, immune systems, you know, are kind of weak and those kind of things. And depending on the culture and the situation, right? Those numbers can change, of course. So bring us back, you know, really until about, you know, maybe 1930, that was almost the case, right? A significant number of children were dying. And then you start to get all these innovations that were popping up, right? Public health measures such as water treatment, huge. flush toilets. That may be the one thing that has saved more people on this planet than anything else, believe it or not, right? Just effective ways to deal with our waste matter, right? So you can see a bunch of other things here, immunizations, penicillin usage around 1945. And so we're doing really well. You can see mortality is just dropping quite dramatically, right? So this is good. We're going to focus in here on the drug usage today, which has saved many, many lives, right? Probably some of us here, right? I know grade 12, I had really bad pneumonia, so I feel that penicillin saved my life. So here's some other numbers, right? So 1900, top 3 causes of death worldwide, right? Infectious diseases, pneumonia, tuberculosis, diarrhea. Bring us up to 2000. Some of these things are kind of gone. Diphtheria, right? You know, does anyone know anyone who's had diphtheria? Not me. My great grandmother died of diphtheria. That's about all I know about it. So it's kind of disappeared due to vaccines and drugs, right? You can see pneumonia, influenza kind of still on there, HIV on there. I believe at least once or twice in the last 10 years, tuberculosis has actually popped up into the top 10. I'd have to find the numbers. These numbers are really hard to find, by the way, from World Health Organization and whatnot. Yeah, so let's talk about those drugs and how they've changed society in many, many ways. So if you go back 100 years or so, right, you know, a little bit more than 100 years, they started looking at the scientific revolution where, you know, people are using scientific method to tackle problems and so on. And eventually, as humans, we start to get a pretty good understanding of some chemistry, right? So early 1900s, you have chemistry as a discipline, and people are starting to be able to synthesize compounds and learn about these things a little bit. So this brings us up to this guy here, Paul Ehrlich, and he had this idea, and he's like, you know what, we've got all these chemicals here, and I wonder if we could find, and he called it the magic bullet, right? So what did he mean by the magic bullet? He's like, if we could find a drug that might, you know, maybe it'll kill the pathogen and have minimal toxicity in the host, right? So not kill the human. So that was the idea. And he got really lucky, right? Only screened 606 compounds before he found a winner. Nowadays, I know people doing this at McMaster. They have this robotic system and people are screening 10 to 50,000 different chemicals for various things. You can't do that without robots. So got really lucky, number 606. And this compound here was discovered. AS is arsenic. So not the best compound. There is some toxicity in humans, but it was effective. Selarsen was effective against syphilis. So this is huge, right? Think about it. People get syphilis, and now you have an incurable disease. Right? First, it's going to start off giving you some genital lesions. It's eventually going to spread to your other tissues. You're going to lose your hair, probably lose your mind in the end, by the time it affects the brain, right? So this is a big deal. I found this on the internet. This is a treatment kit. Kind of looking at it, I'm not exactly sure how it works, but my understanding is somehow they would wash the infected genital areas with the compound, and it's making doctors really rich, some of them, right? People had to go in again and again for these treatments, but it was effective, right? And this, like I said, is a huge deal, right? You know, syphilis now, you're not looking at something that's going to incarcerate you in a hospital anymore. You can get penicillin. So now you've got this idea, right? This idea that, hey, maybe there are chemicals out there that can do this kind of thing, right? So people started looking here and there, kind of starting to get serious. This was another one that was discovered in 1935, Prontosil. This is called a sulfonamide. Some of these are still used today, by the way. Not as common, they're not as powerful as penicillin, but sometimes, you know, when they're looking for an alternate treatment, there's a whole group of these compounds. I'll come back and talk about, you know, what they do in a bit. You can see this is curing streptococcal infections, right? So puerperal fever is an infection that some women get after childbirth, right? You get streptococcus in the blood, and again, and that can also lead to death, right? So now you have a drug that can cure you, and you can get better, and we're all happy. So this is what we want. We want drugs that do this kind of thing, right? So there were not a lot of developments in this era, right? These drugs were effective, but not amazingly so, making some people rich, but you know, I mean, we're not quite there yet in terms of you know, the powerfulness of something like penicillin, which can be used for a whole variety of microorganisms and is super cheap too, right? So let's talk about penicillin. There had been people that had made discoveries like this for decades, but Alexander Fleming was the guy who kind of published and took it to the next level. So remember I said there's this idea in the head about having that magic bullet, having that thing that can kill the pathogen and not the germ, right? So we had to have the germ theory. We had to have that idea in our head that we could find compounds that work that way before we could actually make some progress. So Alexander Fleming was one of these doctors getting rich from salvarsan, right? So treating people for syphilis. And, you know, he had a little lab on the side. He was working in, I can't remember which hospital. This is London, England. I think he was a Scottish guy. He had some sort of cabin. He liked to go there on the weekends. And why is that relevant? Because, you know, he was just getting his experiment ready. And he's like, I'm just going to leave this on the bench for the weekend and they'll come back on Monday, right? And it got an infection, right? So often, you know, when you get mold on your cheese or whatever, you throw it out. In this case, he's got mold on his experiment. I'll show you what, this is not the exact picture, but this is what it might have looked like. And so there's the mold. This mold here is called penicillium. It's actually kind of beautiful, I think. And he's like, hey, would you look at that, right? We've got a zone of inhibition, a zone of clearing. So we're killing bacteria. And he's like, hey, I should maybe take a look at that because, right, going back to magic bullet ideas, and she's treating patients with salvarsan, so he understands the effectiveness of something like this. And so, you know, 1929, he published, he did a few other things, right? So he was not a chemist. So he extracted what he called mold juice. And he figured there was a compound in there, and he called it penicillin, named after the mold. And he found it was effective against some things, right? So it was effective against these, right? A lot of gram positives. Didn't seem to be so effective against most gram negatives. But I don't know why he never tested against syphilis. That's my big question. I don't really know the history of that fully. But this is great, right? We've got something that's killing bacteria, and all of these are pathogenic infections. So, you know, he published, he actually did a couple of other investigations, right? So, you know, he looked at, for example, do I have it on here? Maybe I have it on the next slide. He injected in some animals, no toxicity. I'll tell you, he dodged the bullet here. If he had injected a guinea pig, it would have killed the guinea pig. Would have been game over. Guinea pigs are naturally, it's naturally toxic to the penicillin. He actually treated a couple of patients, right? So somebody had an eye infection and he, you know, I don't know what he did, somehow washed the eye infection with some mold juice, right? And it cleared right up. Somebody with a amputated leg, I believe, had an infection around there too. It worked really well, called it penicillin. But here was the problem. It didn't last very long, right? A couple of days, and then it wasn't very potent anymore. And he's like, well, you know, there's other interesting things. For example, he found a antibacterial protein in his nose. And so that was more interesting at the time. We kind of moved on, right? But lots of people were like, hey, this is interesting. Can we do something with this? So he kind of, you know, he shared his mold samples with people, and lots of people were interested in this curiosity, trying to figure out if there's some way to make use of this, right? So skip forward a few years. Okay, that was 1929. Now we're about 10 years later. I think even more, maybe it was 1940 or so. These two guys here, so we have Florian Chain. Chain was a graduate student, and he read Fleming's paper and was like, hey, can I have some of that mold? And Fleming's like, yeah, no problem. So they brought it to the lab. These guys were chemists, and they were able to extract it and purify it. Here's the penicillin here, the molecule, and they started studying it. Now, what they found is that if you extract it as a dry powder, it doesn't spoil, right? If it's in the liquid form, it only lasts a couple of days. With the dry powder, you can mix it up anytime you want. So they kind of had a system here for testing it. So they started testing it, right? So they're like, okay, let's test it on some mice. injects a lethal dose of streptococci into mice. Mice die, no surprise there. Streptococci plus penicillin, the mice were alive, right? And then what's the next step, right? Eventually you test it on people. So here's kind of the interesting thing when you look in the history of this whole thing around the ethical aspects of this. They did all their initial testing on children, right? You don't usually do that with drugs nowadays. They're more vulnerable. They have more toxicity issues and whatnot. But it actually took a lot of work to make a lot of penicillin. So what's the solution? You find smaller humans, right? Children. But anyway, the good news was it wasn't toxic, right? It wasn't killing anybody. And they were able to start treating some infections. Okay, so this was 1940. What's going on then? World War II, yes. So they realized that, you know, if you look back at World War I, there's a belief that maybe more people died of their infectious wounds than of actual gunfire, right? So this is huge. They realize they have an opportunity to really make a difference for not just human health, but the war effort. So that's one thing they're thinking about. So there was this massive kind of international effort and lots of things to say about that. But one of the things they did is they started getting people to make mold in their basement. Anyone see the irony in that? You know, hey, you want to grow mold in your basement? Oh yeah, no problem. Do it for the war, right? But that's what they're doing. They're actually got a glass company to make these rectangular flasks so people can stack them up in their basements. And you don't do this in a factory because, of course, at the time, England was being bombed. So you don't want one target. You just have tons of people. It's kind of like mom and pop shops of penicillin, right? And it made a huge, huge difference. Eventually, the Americans started making penicillin as well, and they were able to do it at massive, massive quantities. There's all sorts of war posters and things like that that talk about, you know, how this has saved lives, right? Lots of interesting things, lots of stories, right? Of course, you know, they realized that there were a lot of things in the war effort. This was a big deal, by the way, in the war, sexually transmitted infections. You know, you need 10,000 soldiers, and you know, you plan your whatever, and you know, a month later you're like, how much? 2,000 are sick, right? So having that for soldiers was a big deal. And obviously the advertisement started to spill over to other places. Again, things that were incurable, right? And suddenly now we have a cure, penicillin. So 1945 approximately, now you're looking at various companies starting to get into this and mass producing and making huge advances in healthcare. And, you know, to the point where in the 1960s, the Governor General of the United States actually said, we can close the book on infectious disease, right? So I don't know where he got that idea. Look where we're at now. But so 1940s, 1960s, they're like, let's look for more. And so the most common way to look for antibiotics is using soil. So there were people that were going all over the world, you know, okay, a golf course in Japan, right? That's why you went to Japan for the dirt, but then might as well golf while you're there. But all sorts of places, right? Just get little samples of dirt, bring it back to the lab. And so you kind of just spread it on the plate here. And then what do? You incubate it, and then you overlay it with Staphylococcus or whatever, and then you look for those zones of inhibition. And then what do you do next? Well, okay, this colony here, you know, let's just grow that up and see if this microorganism is making any useful compounds. And I think I've got a picture of an actual plate. Like I said, this is a soil plate here. And so they were discovering dozens and dozens of new compounds. Some were actually kind of similar to penicillin, some were entirely different types of molecules and so on. And you're looking at, like I said, a few dozen of these things coming and being very, very successful. Believe it or not, there are still some companies doing this today. Very similar kind of experiments. And every once in a while, they find something interesting. We just haven't dug up all the dirt on the planet yet, right? Here's some examples. It turns out Streptomyces species, these are kind of really funny soil bacteria that are sort of a little filamentous that a lot of people, if you look at them under a microscope, you might think they're fungi, but they're actually bacteria. And so you can see there's a whole big list of various different antibiotics that have come from the Streptomyces. But other organisms as well, we've come up with all sorts of different compounds, and some of these we'll talk about. Some of these, okay, not all of them. Like there's just too many. There's like hundreds of these things if you want to have a good list. So one more definition for you, right? So penicillin, the first one, I don't know why it's called penicillin G and not penicillin A, but the first one is called penicillin G, is a naturally found compound. Nowadays we have chemists, and so sometimes we have synthetic compounds, meaning the chemist has made it from scratch, or semi-synthetic, which means maybe it took penicillin G and we'll add a functional group, or we'll just, we'll just tweak it a little bit, right? So if you see these terms, synthetic, like I said, is completely human created. Semi-synthetic is a little bit of a combination of both. So there's a list of some, like I said, 1940s, 1960s. We still call this the golden age of antibiotic discovery, because this is when most of the drugs were discovered. Still, most of the ones we're using today, or derivatives. Some of them we tweak a little bit, and that gives them a little bit of a longer lifespan. And we'll talk about resistance eventually, but there was kind of a gap where up until about the year 2000, there are not a lot of companies into this kind of thing. And we found a few more things. Again, we're not looking at a lot of companies into this. It turns out that antibiotics, from a business perspective, is not necessarily the best business model for a pharmaceutical company, right? So your average drug takes $1 to $2 billion to develop. Okay, so first of all, you have that. Second of all, problem with these drugs is sometimes you get resistance. So maybe you release a new antibiotic, it's good for 5 to 10 years, and then now resistance organisms are popping up. And you've just blown $1 billion on it. And unfortunately, the last kind of thing, right, with a lot of drugs, this kind of thing, you use it once, and then, your pneumonia is gone, and that's kind of the end. The drug companies, a lot of them have shifted towards chronic diseases, because, hey, it's a lot better if somebody's taking a pill every day for the rest of your life, right? So again, it's a business thing, but there are some companies that are into this. And like I said, there's a number of new drugs that are in development, and it's kind of exciting to see some of this, right? We'll come back to resistance, okay? So one of the things you need to know, right, again, with this history stuff, is this concept here, selective toxicity, okay? So that means the drug is toxic to the pathogen and hopefully minimally toxic to the host. Everything has some sort of side effect usually. Some are a little bit more toxic than others. Unfortunately, some of these things can cause issues with your liver and things like that. And some of them are just outright nasty compounds. But what you want to find is the compound that is sort of perfect, right? So it's not harming the host and is super lethal towards a pathogen. Like I said, there's a few kind of in between. In terms of the number of drugs, we have hundreds of different antibacterial compounds. Hundreds. I don't know how many hundred, maybe 200, but something like that. And we have very few antiviral drugs, maybe a dozen. Maybe a few more now, right? With AIDS and coronavirus, there's been a lot of development in Ebola. There's a bunch that have sort of popped out. So let's say 15 to 20. So what is going on here? Why do we have so many antibacterial drugs? It has all to do with biology, okay? So a bacteria is very different than your cells. So if you think about all the biological differences, they have peptidoglycan. They have a cell wall that is unique to bacteria, and that's a great target. If we can find something that can disrupt the cell wall, we can kill these things. The ribosomes are different. They have different DNA enzymes. They have all sorts of things that are very different. Viruses, on the other hand, they're using your enzymes. So you don't want drugs that are going to inhibit your enzymes. So it's a little trickier. These guys, so fungi and protists and parasites are somewhere in the middle. They're a little bit different from us, but they're still eukaryotes. So it's a little more challenging to find some products that are going to inhibit them and be selectively toxic. Okay, so bacteria are kind of the big one. So we're going to spend the bulk of this talking a lot about antibacterial drugs. Okay, plus they're kind of the big one, right, in terms of the effectiveness and the broad usage as well. So we're going to kind of talk about 5 different types, or at least 5 different drug targets. So I mentioned that the bacterial cell is different from the human cells. Here are the most common targets, and some of these drugs we're going to talk about, a few others we're just going to ignore, but you can see there's a list on here. So the cell wall is kind of a big one. because it makes them really vulnerable. If you put holes in their walls, they leak and they die, right? So that's a really good one. There's also drugs that affect the plasma membrane. There's some differences, right? Particularly gram-negatives. Remember, the gram-negative outer membrane has lipopolysaccharides, which is a totally weird thing when you compared to us. Ribosomes are different. I mentioned DNA replication, and sometimes there's other pathways. So I'm going to spend a bit of time talking about the penicillins in particular. So let's talk about the penicillins here. So like I said, 5 categories, right? And tons of examples in each of these categories. So I think I have 1 example or 2 for every category that you need to know. Okay. So in many cases, I've tried to put them in bold, the ones that you need to know bold, but I'll try to point them out. So let's talk about the penicillins first. Okay. So how do they work? You have these enzymes and they build the cell wall. They're actually called penicillin binding proteins because we didn't know what they were when they were discovered, right? Other than they bind penicillin. So we had no idea what they were doing. That's actually a huge part of my PhD research is on penicillin binding proteins. So these things and other enzymes, they kind of go along. And if you remember peptidoglycan, there's the glycan part, that's the carbohydrate, and there's the peptide part. So they're kind of going along and they're cooperating. And, some of them are building new carbohydrate bonds. Some of them are building new peptide bonds. And when you put penicillin in there, it inhibits those penicillin binding proteins, and that affects these cross-links. So by cross-links, I mean peptide bonds that are linking the 2 different carbohydrate strands, and these holes are left, right? And so that's what happens. You get holes. I have a little video I'll show you of some bacteria that are dying here in a moment. Penicillins are also called beta-lactams. I'm going to use the term penicillin or penicillins because it's a group of drugs. The chemists like beta-lactams. Both are interchangeable. Most people remember penicillins better than beta-lactams because this is a chemistry term. And so if you're familiar with the chemistry, great. But most people are not familiar with that kind of unique structure. And I'll point it out here in a minute. So they lead to holes in the cell wall. Got a video here I'll show you in a second. Here's kind of another little animation I made for a presentation one time. But there's our cell. And it's kind of funny when you think about it, why are they doing this? As they grow, they're putting holes in their cell walls that they fix up. And it kind of makes sense. If I was like, okay, I want to expand this classroom, let's say another 5 meters in that direction. First thing I got to do is get a sledgehammer and start taking out the wall, right? And maybe parts of it I'll push back and then repair or something like that. And that's kind of what they're doing. They fill in the new materials and then they divide, right? Here's my penicillin in there. And there it comes. It's like a wrench in the enzymatic machinery. And so the cell just, right? So that's my little animation. But I know a video here somewhere. There's the video. Let me play this video for you. I hope it plays. So watch carefully. They're going to stretch and then they're going to go. Can't remember if this one has sound effects, but I used to have a really nice one with sound effects. They would make explosion sound effects, but I don't think this is the one. I'll play it though. So they're stretching out and then watch, they're going to just suddenly go, there we go. That one went, that one. There they go. So penicillin is really effective on vegetative cells, growing cells, right? If you have endospores, so Clostridium difficile, C. diff, right? Not going to be effective until they start growing. But that's kind of the thing, right? And we'll talk a little bit about that a little bit later on here. But there's penicillin. I've got a whole bunch of penicillins here. I actually have penicillin right here. There we go. There's penicillin. Made this model a long time ago. But one thing to point out is this little square ring, which I have highlighted on here. That's the beta-lactam ring, right? That's where the term comes from. Some chemist named it, and now the name has stuck. But often it has this sulfur in it, and you can see my sulfur here. It's the yellow on the structure, right? So black is carbon, blue is nitrogen, red is oxygen. There's usually a set color for these things on the models, but have that sitting around in my office, so I like to bring it out once in a while. It's nice to have toys. There's a bunch here. Like I said, there's a huge. This is maybe the biggest group. So if the antibacterials are the biggest group, the penicillins are the biggest group of the antibacterials, and probably 100 of these. Right, maybe 150. I'm not sure. Lost track. A lot of them have "in" on the end. I don't know why they have that. Some of them are penicillin. There's the original that was discovered by Alexander Fleming. We don't use that anymore. You can see the issue. It says it's injectable, right? So much easier to give someone a pill, right? If you need a drug, it's not the best to have something that has to be injected, because that means they have to go to a clinic or hospital or some other healthcare facility. So if we can avoid that, this is the good one, right? You can see it says it's acid resistant. So this one here is the one that I think saved my life. Remember when I was a university student, I had pneumonia in grade 12 and in university, unfortunately. My drug plan, I think it would pay for the drug, but then you had to pay the dispensing fee. So get this receipt, right? The dispensing fee was $16. So I paid that, and you get the receipt, and it tells you that their drug plan paid $4.25. Right? This stuff is dirt cheap, still used today, particularly if they don't think you have any organisms that have resistance to them. That's one of the issues with this one here. Yeah, it's been around a long time, so there is resistance to this. This is where ampicillin came from. This was made semi-synthetically, so you can see if you look at the structures, they took penicillin G and they kind of tweaked it over here, right? And it turns out this one here is a little bit more broad spectrum. We'll talk about broad spectrum in a minute, but it means it hits a wide variety of organisms. Penicillin G, right? Gram positives, E. coli not so much. Ampicillin, right? Gram negatives as well. So bit more broader of a range of what it attacks. There's a few others here. You can see this one's called the cephalosporin. Don't worry about that term. It's just a bit more broad spectrum, also semi-synthetic. Methicillin, we're going to talk about a little bit, right? Methicillin resistant Staphylococcus aureus. This is a big deal because when it came out, I think in the 1980s, it was amazing. It worked well. And in many cases, it was the drug of last resort, meaning that nothing else worked, but methicillin will work, right? Until suddenly we have resistance. And that's why methicillin is a big deal, right? Particularly methicillin resistant microorganisms. So that's penicillin, okay? You better know what penicillin does for the midterm and the final exam, okay? So that's not a hint, it's a promise, okay? There will definitely be a question on it, okay? I've had the question, right? Why should you not give penicillin to somebody who has the flu? And the right answer is because the flu is caused by a virus and penicillin will be ineffective, right? If I were to give you one question, that would be, you pass the course or fail the course, that would be the question, right? I'm not allowed to do that, by the way, but that would be the question. I think it's one of the most important questions when we're talking about drugs, right? In terms of having an appropriate drug. And, as healthcare professionals, you're going to see people that are demanding antibiotics when they have viral infections. People will demand them, and you have to say no. Okay, it's very important. One other I want to talk about, like I said, there's a bunch of drugs that attack the cell wall. Some of them are using other mechanisms. We're not going to go into all of them. There's one other that's really important to mention. This one here, isoniazid. Okay. And you may remember that term, mycolic acid. This is found in tuberculosis. So Mycobacterium tuberculosis has that waxy mycolic acid layer, and probably no surprise, it's resistant to a lot of drugs because they can't get past that waxy layer. Okay, so hey, let's find a drug that inhibits that mycolic acid synthesis, and that's what this does, this isoniazid. So there's our acid fast stain, right? I thought I had another slide. Maybe that's it. Maybe this is it. So just this is like one of these, we don't have enough tuberculosis drugs, unfortunately. This one here is still used. A lot of cases we're looking at drug resistance and tuberculosis. So when we treat tuberculosis nowadays, it used to be standard that if they thought you had normal tuberculosis, meaning not multi-drug resistant, they were giving you this. I think now the standard of care is they're always giving cocktails now, meaning mixes of drugs, because the resistance is just really common and they're worried about, if you hit it on, you attack the microorganism at different fronts, but this is pretty much in all the cocktails. So it's a good one to learn. Okay, so that's the cell wall. What other targets can we look at? We can look at the membrane. Remember I was saying the gram negatives have unique membranes, that outer membrane. Polymyxins fit that category. I love chemical structures of antibiotics, by the way, because I look at these chemicals and it's just like this is crazy, right? That these microorganisms are producing these compounds that are just like, wow, look at that thing. So kind of a cool compound. So polymyxin B is one of them, one of the most common ones. And there's our outer membrane, and there it goes. It gets in there, and it's making that membrane leaky. And of course, a leaky membrane is bad for the microorganism, and it's going to kill it. Now, this here is not the best compound. You're not going to take this one orally because it does have toxicity in humans. We also have membranes. They are different, but it still has enough toxic effects. But you see this in a lot of products, right? Any topical products, anyone know with the word poly in it? Yeah, it's like half of us, right? When we were little, you know, so I remember my mom was like, oh, you got a scratch, polysporin, right? You know, I'm sure lots of us have been in that category, but there's all sorts of these on the shelf. I was taking a look, polysporin, like there were like 6 products there in Shoppers Drug Mart. But if you take a look, you can see the active ingredients. It says polymyxin B sulfate, and then there's a second antibiotic, one that we're not going to talk about here. Oh, there it is, back tracing. Neosporin has 3 different topical drugs. So I don't know, but then I found another polysporin that had 3 topical drugs in it. So like I said, I don't really know why there's so many, but I think it's just product placement. The more products you have, the bigger chunk of that shelf you get, right? But you see these are really common type of antibiotic creams. And you know, if you have an infection, you know, it doesn't hurt to stick it on the Band-Aid and might help you out. Okay, so the other group that may be the second biggest group are drugs that target bacterial ribosomes. And there's a huge number of these different compounds. I think the textbook covers about 4 or 5 different categories. So there's cephalosporins, gentamicin, a bunch of other compounds. There's 2 that I want you to know. I'm going to show you 3, one just because it has historical importance. But these things bind specifically to bacterial ribosomes. So remember what a ribosome does? Okay, it makes proteins, so it takes that messenger RNA and then it strings together some amino acids. Okay, so if you read kind of the details in the textbook, it's going to tell you, you know, some details about what's going on. Like some of them actually block the messenger RNA, some of them bind to the large subunit and block amino acids from arriving, and different things like that. I'm not going to get into all the nitty-gritty details, but I'll show you kind of basically what is going on. So remember, our eukaryotic and prokaryotic ribosomes are different. Our prokaryotic ribosomes are actually smaller, and their composition is different. So again, this is perfect because you have a target that a compound, if it binds really well to this guy and not very well to this guy, and awesome, right? We've got a drug. And it turns out there's a whole bunch of them. Okay, first one to know, let me just check where we are with time. Okay, lots of time. First one to know are the macrolides. So they actually bind the large subunit. Here's one here. Again, like I said, I love these compounds because these are the kind of things, you show an organic chemist these compounds, they're like, how on earth does that microorganism synthesize that? That's a nightmare to synthesize in the lab, but you can find them naturally. So this is one to know. Erythromycin. Okay, this is really commonly prescribed in situations where people have allergies to penicillin. It's kind of the go-to drug. Oh, you have a penicillin allergy, we'll give you erythromycin. It's actually a really broad spectrum drug, meaning it will treat gram negatives and gram positives and a bunch of things. So used for all sorts of things, right? Bladder infections, skin infections, you name it, right? Huge list. And like I said, one of the key things is, you know, if someone has a penicillin allergy, this is totally different molecule, right? We don't have that beta-lactam ring. So usually, if you're resistant to penicillin or allergic to penicillin, you're not allergic to this one. Okay, so erythromycin is a really important one for that reason. There are others. That's just the one I want you to know for the class. Okay, like I said, I'm going to give you one example in each category. There are some of these drugs that bind the small subunits. So this one here blocks transfer RNA docking. And so different mechanism. You can see it's a different chemical structure. And this is also a really strong broad spectrum antibiotic, meaning it's very good at killing a variety of bacterial species. There are some issues with this. We don't usually give it to children. I'll show you why. Because it is known to stain the teeth. I used to work with tetracycline. It is a yellow compound. It comes as a yellow powder. And so I don't know why it binds to growing bone matter, but it does. And so there are people out there that were treated with tetracycline as children, and they have yellow teeth. I Googled it one day, and there's a few dentists out there that have treatments for that. I don't really know what they're doing with that. But like I said, very strong antibiotic, works against Pseudomonas. Remember, Pseudomonas was the one that, not everything kills. Effective against Pseudomonas. So this is a really strong one. Not always the first thing prescribed, but if somebody has something that's difficult, tetracycline is kind of one of those go-tos. Although there are other tetracyclines, it's actually a group, right? There's tetracyclines. There's a few different related compounds. I told you I'd show you one more, important for historical reasons. I won't ask you about this one on the test, but streptomycin was the first anti-tuberculosis drug. So this one has huge historical significance, right? 1920s, you had tuberculosis. What did they do? They put you in a sanatorium, which is a hospital for people with tuberculosis, and they hope you get better, right? Sometimes you're there for months, sometimes you're there for a couple of years, sometimes you don't come out alive. Streptomycin came out, now there's a treatment, right? So you can see it says here there are some side effects, right? This is why we don't use it anymore. Some cases as a last resort, but it can cause ear damage. So some of these things, our ribosomes are not that different between bacteria and humans. That most of these drugs, if taken too long or in high concentrations, do have human toxicity issues. And there's an example of one. Why the ear? I have no idea. I don't know why the ear versus other tissues. I don't know enough about streptomycin. Okay, so two more categories for bacteria. Category D is the quinolones or the fluoroquinolones. Fluoro means fluorine. That's one of the atoms on the periodic table. This one here came out in 2001. And at the time, this was a brand new type of drug. So there was no recorded resistance because it had never been prescribed before. So it was a big deal when it came out as kind of a new generation of drug. We do have resistance now. Again, another strong broad spectrum drug, works really well. Probably is overprescribed because you're starting to see a lot more drug resistance on these now. But again, you've got a different mechanism. So sometimes that's what you have to do. They might have resistance against beta-lactams, so you hit it with something that has a completely different mechanism. So what does this do? I'll kind of show you here what's going on is when you replicate DNA, you've got a twisted double helix structure, right? So, if you have a rope or a hair braid, you don't just yank at it and pull it, it's gonna knot up. The same thing with DNA. If you just pull it apart, it's gonna knot up. So these enzymes actually they untwist, untangle, right? What they actually do is they clip the DNA, let it untangle, and then reform it, right? So this analogy is not the best because nobody has a phone anymore, but I have a phone in my office, and I don't know why it does, but that cord always gets tangled up. So I unplug it, let it untangle, and then replug it. And that's what these enzymes do. There's two of them, topoisomerase and gyrase. Like I said, they're untwisting type of enzymes. And they're unique to bacteria. We have different types in eukaryotic cells. So there we go. The fluoroquinolones are inhibiting those enzymes. Okay, so I know your brains are kind of getting on that full edge. Then one more antibacterial, and then we'll have a little bit of time to talk about resistance and a few other things. There's sort of the fifth category, which is targeting other, it's kind of that other category where you're targeting things that are unique to bacteria. So folic acid production is one. So Folic acid, you're probably thinking, hey, I know that. That's vitamin B9. I like vitamin B9, right? It's a vitamin. And by definition, a vitamin is something we have to have in our diet because we don't make it ourselves. So you have to eat it in foods, right? So that means we don't make it. Bacteria, on the other hand, that's not what they do. They make their own folic acid. So if we can stop them from making it, then it's going to kill them because we actually use this to make nucleotides, right? So can't make that. You can't make nucleotides. Humans get around it by just eating it. I'll kind of show you what's going on here, right? So here's our kind of part of the metabolic pathway to make folic acid. There's this intermediate here called PABA. You can see a whole bunch of steps. Eventually, folic acid is made, and eventually nucleotides, right? So the sulfonamides. So this is the one we were talking about way back at the beginning that is for treating streptococcal infections. Basically, it kind of looks like this PABA molecule, right? So it's similar enough that it binds the enzymes, and the enzymes are fooled. They grab onto it, but it kind of sticks to them, right? It has a high affinity for them, and it inhibits them. And so now the folic acid production comes to a grinding halt, and the microorganisms they can't make nucleotides, so then they die, right? So there's a whole bunch of people kind of targeting unique pathways in bacteria, hoping to find the next big thing, and maybe we will, maybe we won't. I don't know. It takes time. Like I said, $1 billion to develop a new drug, and it's getting more and more difficult to develop antimicrobials, unfortunately. Lots I could say about that, but we're gonna move on here. I want to talk a little bit about resistance today. That's probably going to finish where we're going to get to today. So here's some of the examples. I think that is all of them. So this is the slide, right? You know, the ones you need to know slide. Maybe I should put a little star on it right there. You know, boom, boom, boom. You know, don't forget these ones. Okay. I think last year it was a matching question, right? So, you know, I had the drug on one side, a description of either the mechanism or something else about it. Right, I think maybe for erythromycin, you might say, might be prescribed if someone is allergic to penicillin, right? So maybe some other detail like that, right? Okay, I know it's scary, right? But wait till you take pharmacology. We've got about 1,000 drugs to learn then. So we just have just a few, right? So one of those things, you know, you get back to the end of the semester, right? I know the organic chemistry students were talking about, you know, there's really only 7 reactions to know, right? But you did the whole course and it's just like, holy cow, is that it? Like that's nuts. But like I said, a small list here. So I just want to talk a little bit about broad spectrum versus narrow spectrum. Okay, so broad spectrum, there's some examples, and will inhibit many types of bacteria. Narrow spectrum, more limited activity. So maybe it just attacks gram positives, or maybe just salmonella, or something like that. So why do we care? These are useful because sometimes you may not necessarily know what the patient has. Excuse me. So it's useful to have a broad spectrum drug. There's lots of cases where somebody might have pneumonia, you give them the standard treatment, it doesn't work. So you throw a broad spectrum at them because it's like, this person's sick, we want to do something about this, right? So broad spectrums have their use. Unfortunately, they kind of just wipe out a lot of stuff on your body, right? You're talking about wiping out your intestinal flora. There's lots of good bacteria in and on your body. Sometimes this leads to, we talked about the antibiotic-induced diarrhea with Clostridium difficile, right? So we don't want that. So sometimes that happens. Sometimes when people take broad spectrum drugs, can lead to yeast infection in women because yeast are not bacteria, right? So what you've done is you've wiped out a big chunk of the bacteria, and the yeast have a chance now to thrive and can cause a yeast infection. So this is why, you know, we like the narrow spectrums, but sometimes they just don't work, right? So, you know, we're trying to prescribe these generally, but these in some ways have their use, and, you know, so it's that balance of trying to, you know, treat the patient best and not overdo with the broad spectrums. So they have their uses, okay? That's kind of really what you need to know. I got a slide about this. Advantages, kill many types. No need to wait for culture results. Like I said, sometimes the first treatment didn't really work very well. Disadvantage is the effects it might have on your microbial flora. Okay. Have anything else there? Yeah, and in many cases you're looking at opportunistic infections as a side effect of that kind of thing. I don't really know a lot about this, but I do see sometimes every once in a while somebody shares their drugs with me, and I take a look at them and try to understand what's going on. You know, there's somebody who had some sort of acne, and they were prescribing tetracycline. Again, I don't know enough about acne, but it just seems to me that there was a mismatch there. Tetracycline is very broad spectrum. It's very good at wiping out half of your flora very easily. And I think there were risks to having that. Again, it may have been a point where they had it, other treatments were not working. I don't know all the details there, but you do see that kind of thing relatively frequently. Okay, time check, just past 2 o'clock. Okay, we'll talk about a few more things. We'll wrap up eventually here. A couple other definitions aren't so important. I'll never ask you about this on the test, but I know in the textbook it's using these words a lot. Just wanted to define them. Some drugs are bacteriostatic, some are bactericidal. So some is killing them, and some is just sort of stopping them from growing. It's kind of all you need to know. Usually both are effective at treating infections. If they're not replicating, that's kind of the main thing. The infection doesn't have a chance to propagate. But yeah, I know the textbook uses these words, drops them a whole bunch of times, and there are advantages and disadvantages. We're not going to get into that in this course, but there's the definitions. Won't ask you on the test about them. Okay, so how do we test the effectiveness of these things? I told you basically same idea as the disinfectants. You can do this with antibiotics. You can actually buy these discs, right? This one here, gentamicin, chloramphenicol, tetracycline. Maybe you can see it's a little yellowish. What do we have here? Ampicillin, kanamycin, penicillin B. You can actually buy these standard discs, and you can, you know, you might have a clinical isolate, and we want to see how effective this drug is, right? So, you know, just get swabbed on a plate, throw these little discs down, and help to determine which treatment is going to be most effective for that particular isolate, right? Same test, just it's antibiotic instead of a disinfectant. These numbers actually are the concentration, so 30 micrograms per mil, for example, for the kanamycin and so on. You want to be a little bit more quantitative. You're going to measure things out in the test tubes, right? So minimal inhibitory concentration test. You just do these dilutions. I did this a bunch of times, and you can see usually what happens is you've got growth here at the low concentrations. So this is 8 micrograms per mil, or sorry, 0.8 micrograms per mil, and it still has growth. You can see right here, 1.6 micrograms per mil, and it's effective, and that's the effective concentration, right? So sometimes this information is used to for dosages, right? You want to give a person a certain amount to put in a pill, and you know, thinking about body size and all those kind of things in terms of having an effective therapeutic concentration. A lot of these drugs, you know, 1 or 2 days worth of use, the effective concentration isn't built up enough, and this is what they're trying to do with the standard treatment, is to get that effective concentration to a certain level. So don't want to do a disc diffusion, or you want to do an MIC test, but it takes too much work. So somebody invented this e-test, which is kind of like the best of both worlds. It's easy like the disc diffusion test, but it's going to give you quantitative values. And so this e-test, you get this little, I don't know what you want to call it, a stick, little band, and it has actually different concentrations embedded in it. And so you can read right off the band what the MIC is. It's not quite as accurate as doing the test tubes, but this is quick and easy and takes a lot less time. You can get kind of an approximate number. You can see in this one here, they're comparing, looks like penicillin and erythromycin. So effective, both are effective against this particular microorganism. Very easy to do, and you get the advantage of the quantitative test. So that's good. We like that. We like easy. We like to have numbers. Okay, so that brings us kind of back to here, full circle, right? We've got all these great drugs. Not much is going on for a while. unfortunately, we have some resistance, so we have to talk about that a little bit. I'm not sure how much we'll get into today, but innovation gap, right? That's a big deal. So let's talk about resistance, right? So picture Imperial March here. What is going on? So these things have been living for a long time. Many of these things are in a soil, a teaspoon of soil, right? Healthy fertile soil. You're looking at like probably 10 or more thousand different species, right? So this is just basic biology where you have all of these things that are living in the same ecosystem and you're competing against all of these other species. So when you're small and you're a microorganism, how are you going to do that? You're going to secrete chemicals and hopefully that will kill your neighbor, right? And then you get the food and they don't, right? That's what they do. It's kind of like warfare. And so what do you do if there's another species in the soil that is producing a chemical? You can develop resistance against it because you're going to survive, right? And so this is a sample of thought it was soil. I can't remember what it is, but you can see all the different species in there trying to compete for that limited amount of space and resources. Tons of studies like this. This one is from not our textbook, but one of the other ones I have on my shelf. And you can see this is looking at drug-resistant gonorrhea. So 1980, almost non-existent. By 1990, almost 10% of cases were drug-resistant, right? Tons of studies like this with different antibiotics and different microorganisms. And all the trends are showing the same thing, that resistance is on the increase. So it's not necessarily if it's going to happen, it is going to happen. It's just, are we going to be able to put this off for 5 years or 10 years? These organisms do find ways to find resistance. And we'll talk about some of those mechanisms here in a minute. How do they resist? Kind of worth talking about for a minute as well. This is kind of a little sketch. I'll show you what's going on here. A lot of this is kind of some biochemistry that, we're not going to get into the details, but kind of just showing you what they're doing, right? So one thing they can do is destroy the drug, right? So that makes sense. The drug gets into the cell and they have an enzyme that, is like Pac-Man, just eats it up, right? Chops it up. So there's an antibiotic degrading enzyme. Pretty common for certain types of penicillin resistance. Sometimes they alter the drug. It doesn't destroy it necessarily, but maybe blocks it, adds an extra functional group on it, kind of a similar category. Another method is these efflux pumps. So some microorganisms, they have these sensors, and if it's something that they don't like, they're like, this isn't food, they just pump it out, right? Kind of seems really like energetically wasteful, but hey, it works. It's really effective, these efflux pumps. I think I've got some of the examples here, right? These are, I was trying to limit it to mostly the examples that we talked about. And you can see there's multiple mechanisms of resistance for pretty much all of these drugs found in the microbial world. So this is a big concern. I think the real question is, how do they get it? So here's a little cartoon for you that kind of explains part of it. How did they get resistance? You can see it says here, it was on a shortcut through the hospital kitchens that Albert was first approached by a member of the antibiotic resistance, right? So you can see the shady character here, and he says, hey, kid, you want to be a superbug? Maybe he's giving a little bit of DNA, right? So it's all in the DNA. That's what we need to talk about here for a couple of minutes, and then we'll wrap this up. So there's kind of two mechanisms, right? Scientists call these horizontal and vertical transmission, right? So in terms of biology, usually when we say vertical transmission, we're talking about generational stuff, right? So maybe you inherited your, beautiful blue eyes from your parents, right? That's a vertical transmission. You're inheriting that from your parents. So take a look at this. You've got some microorganisms there, and somebody has they're sick, and they're not taking all the pills they should, so they're at kind of sub-therapeutic doses. And you've got this one guy who's just got a little mutation. So mutation is a change in the DNA, and this happens, right? These microorganisms, they grow and they can have little mutations that spontaneously happen. So once in a while, you get a mutation that helps them out. They're just a little more resistant. And so the drug kills these lighter colored ones, and this one survives. And so what does it do? It divides and reproduces And now you have something that is drug resistant. And I'll show you a terrifying video here in a moment. Absolutely terrifying, just in time for Halloween, right? So I'll get to that in 1 second here. It might be on, it's either one next slide or 2 slides from now. Yeah, so selection, right? You can have, like if you think about biology, natural selection is, all that survival of fittest stuff. And this is the same idea, right? So the living ones are selected there, right? And then the other ones die. Yeah, this is natural, artificial. I don't know where you call it, so I'll just use the word selection. There are other ways to obtain resistance by having DNA delivered to you, right? So why go through the mutations if there's some other organism out there that can already do it, right? So there's different ways to get this. This is called horizontal, and I'll show you some of the different ways. Some of them get it through conjugation. So we were talking about that, right? This is that bacterial sex where they have that pilus, and it's like a grappling hook, and they give the recipient some DNA, and sometimes on that plasmid is a gene or two for drug resistance. So that's one way to do it. They don't have to be the same species. That's why I made that one a different color. Sometimes there are other ways. Some microorganisms, gonorrhea is very good at just finding DNA in the environment and trying it out, right? This is called transformation. And this happens all the time, right? Microorganisms are dying and there's just DNA in the environment. And like I said, gonorrhea is very good at just, hey, there's some DNA, grabs it, samples it. If it's of some use to that organism, for example, resisting drugs, it survives, and that's bad for the person who has the gonorrhea, of course. In some cases, we have some examples of transduction. So this is bacteriophage that are carrying DNA, and they infect a microorganism. Sometimes they have a gene, doesn't kill the recipient cell, and now it's genetically modified. So these things here tend to be a little faster. So you can see I have some little nastier looking bugs here. Some of them are super bugs, which means they have resistance to multiple drugs as well. Okay, so I have this video for you here. And this is where I'll finish off. Video unavailable. Let's try searching that. Try that again. I don't think that's it. I guess there it is. I don't know if there's volume here or not. Let me just give me another second here. I'll see if I can figure this out. Okay, I'm gonna have to just let this play and tell you what's going on because I can't figure out how to put the volume on. So what this guy did is he built this massive petri dish. So a giant rectangular petri dish, right? Is it moving? Yeah. So what he does is he has these different zones of antibiotic concentrations. So you can see he's actually the closed caption will be explaining it. So the zone on the outside is 0, right? And then 1 times the therapeutic dose, and then 10 times the therapeutic dose, and then lastly 100 times the therapeutic dose. So then what he did was inoculated the petri dish, right? And so we'll watch what happens here in a moment. So there it is, and you can see the bacteria very quickly grow in the zone of 0 antibiotic. And they keep growing, and then eventually you have a mutation. Right there on the right, you can see some resistant microorganisms, and now they're growing, and they're going to take up that space of 1 times the therapeutic dose. Scary thing is they don't stop. 10 times they start to migrate into that zone. So what's next? 100 times. So like I said, this is just absolutely terrifying. Now, not all microorganisms can do this so readily, and this particular drug is something that it's easy to get mutations for resistance. But you get the idea, this selection kind of thing. And this is what's happening when drugs are being abused. If they're being overused, for example, it used to be legal in Canada to give antibiotics to livestock. So just feed it randomly to your chickens or your cows or whatever. And so what are you doing? You're overusing drugs and giving opportunities for these things to evolve resistance. Sometimes people don't take the whole 10 days of dose. And so you have subtherapeutic amounts in your body. So when they tell you to take all the pills, they really do mean it because they want you to not have the microorganisms to have resistance evolving in your body. Because if you do that, next time you may be out of luck when you have that same infection to have the same drug. So that's where I'm going to finish today. We'll come back and talk a little bit more about resistance next day. Thanks for coming. We'll see you on Friday.